Greetings to everyone. Welcome back once again to my educational channel on biology. In this video, we're going to discuss activity 5.2 from the Form 4 Biology Case Exam syllabus. The objective of this experiment is to study the effect of pH on the activity of the enzyme pepsin. Okay, effect of pH. pH is our manipulated variable on the activity of the enzyme pepsin. So activity of enzyme pepsin is our responding variable. So we prepare 3 test tubes P, Q, and R and fill them with the albumin and pepsin. Okay, albumin contains a type of protein, pepsin is the enzyme, all right, and albumin is the substrate. So all these 3 test tubes have albumin and pepsin, but there's one difference, and that is the pH of the reaction medium or the mixture. So in test tube P, we put in HCl to create an acidic condition. For Q, water is added to create a neutral condition, that is distilled water. And for R, we add in sodium hydroxide to create an alkaline condition. So the 3 test tubes are immersed in a water bath at a temperature of 37 degrees Celsius and left there for 20 minutes. And the condition of mixture is observed at the beginning of the experiment and after 20 minutes. Okay, so whether the mixture is cloudy or clear. So what is the end result and what actually happens in this experiment? What's the reaction that occurs in this experiment? Let's find out more. Okay, and we'll also be discussing the technique of answering paper 3 questions in the later part of this video. So we are going to use albumin or egg white. Okay, it's taken from the white of the egg here. It is made up of 90% water. Albumin is made of 90% water and 10% proteins, including the protein called albumin, M-I-N, but the egg white is called albumin. Okay, so usually in the lab, the albumin comes in the form of a powder. It's dried form, and then a suspension can be made using the albumin powder, or egg white from the fresh egg may be used instead. Okay, but not the egg yolk, just the egg white. So when you put egg white or albumin into water, it will form a suspension, a whitish or cloudy suspension. Now, what is the reaction for this experiment, 5.2? So we're going to put in enzyme pepsin and mix it together with the albumin. After some time, what should happen? At the right or optimum pH, such as pH 2, hydrolysis should occur, okay, whereby the protein is broken down into polypeptides. So the protein in the albumin is broken down to form polypeptides when hydrolysis occurs at pH 2, okay, and this action is carried out by the enzyme pepsin, which breaks down the protein into smaller molecules called polypeptides. So the protein is the albumin, which is a cloudy suspension. Now, after all the protein in the albumin has been broken down, the cloudiness will disappear. So, because polypeptides form a clear solution, all right? So after about 20 minutes, the cloudy solution should become clear if there is digestion of the protein or the albumin. Okay, let's look at this picture here. All right, so originally it's quite cloudy, all right, but if there is digestion or hydrolysis of the protein, it should turn clear like this. Slowly it should turn clear, right? Means you can see through the test tube even though there's a solution inside, just like when you pour water into a test tube, right? So this is the clear condition because all the protein has been digested to form polypeptides, okay, which are soluble in the water. So it forms a clear solution. Now discuss activity 5.2, and the objective here, or the aim here, is to study the effect of pH on the activity of the enzyme pepsin. So the effect of MV on RV, okay, usually it's the effect of MV on RV. So effect of MV, the pH here is the MV, right, on RV. So activity of the enzyme pepsin is your RV, okay. Now, after you have identified the MV and RV in any objective, then you can write down the problem statement, hypothesis, and so forth. So the problem statement should include the MV and the RV too, okay? And then the relationship is in a question form. For problem statement, there's always a question mark at the end, all right? It is actually a question. So the problem statement is, what is the optimum pH for the most efficient or the fastest reaction of the enzyme pepsin. So I prefer you to have this word most efficient or fastest reaction, which is not found in the textbook. Alright, but this is needed to form the relationship, okay, between the word optimum pH and the most efficient or fastest reaction. So our problem statement is, what is the optimum pH for the most efficient or the fastest reaction of the enzyme pepsin? Just write both down, okay? So the optimum pH, the word pH here is your MV, right? And then RV is the reaction of the enzyme pepsin, and your relationship in question form. Now, optimum pH for the most efficient or fastest reaction. That is that relationship, but it's in a question form, right? Now, hypothesis is a statement. So now hypothesis will answer the problem statement, okay? So the hypothesis here is that we need to prove in the experiment is that pH 2 is the optimum pH for the most efficient or fastest reaction of the enzyme pepsin, alright? So again, check whether you have your MV, RV and the correct relationship, which is what you should have for hypothesis. Now, MV is your pH, RV is your reaction, the reaction of the enzyme pepsin. And your relationship is the pH 2 is the optimum pH, optimum, for the most efficient reaction or fastest reaction, okay? Now, coming to variables, our manipulated variable is the pH of the reaction medium and also the pH, yes, the pH of the reaction medium, okay, correct. Now, our responding variable is the rate of reaction of the enzyme pepsin. So this is connected to your main RV here in your objective, okay, or in your problem statement. The reaction of the enzyme pepsin is the responding variable, but here to measure, we just write down rate of reaction of the enzyme pepsin, okay? That is the main RV taken from your problem statement, hypothesis, and also from your objective especially. But in order to measure the rate of reaction of the enzyme pepsin, we have to observe the condition, which is the clarity or turbidity of the mixture of albumin and pepsin, okay, at 0 minutes and after 20 minutes. So this becomes your sub RV or your operational RV, which you use or you detect or observe in the experiment. Okay, after 20 minutes, you should observe the condition, that is the clarity or turbidity of the mixture. Now, clarity means how clear it is, and turbidity means how cloudy it is. Okay, so write both down, the condition or the clarity or turbidity of the mixture. Okay, I think textbook also use the state of the mixture. Okay, the state or condition of the mixture, or the clarity or turbidity of the mixture. Our fixed variables, there are many fixed variables. Firstly is the albumin concentration. Next is the pepsin concentration, which is 1% solution. And okay, so here is written pepsin solution concentration. And then the temperature of the reaction medium is kept constant at 37 degrees Celsius by using a water bath. Okay, so 37 degrees Celsius is the optimum temperature for the most efficient reaction of the enzyme. Okay, so it's kept constant because our manipulated variable is actually the pH in this experiment. Okay, so fixed variables. Now let's go on to the materials and apparatus, and we have a look at these materials and apparatus on the next slide afterwards. So materials are albumin suspension, that is the egg white. Now the egg white usually in the lab, it comes in the a powdered form, okay, or the teacher may even prepare a suspension of the egg white from the fresh eggs, alright? Sometimes it's even boiled so that you can see the proteins, the white proteins more clearly. But never mind. So normally it's a solution made, a suspension made in the lab, okay, using powdered albumin. 1% pepsin solution, 0.1 m hydrochloric acid, 0.1 m sodium hydroxide solution. So all these two reagents are to create the right pH of the reaction medium, okay, that we need. Now, water bath at the temperature of 37 degrees Celsius, pH paper, and distilled water. Apparatus, we need a beaker, dropper, thermometer, test tubes, okay, test tubes in a test tube rack, 5 ml syringes without needles, stopwatch, Bunsen burner, tripod stand, wire gauze. Now, Bunsen burner, tripod stand, wire gauze is for the Bunsen burn, is for the water bath, okay, and then thermometer. Materials. So to study the effect of pH on the activity of pepsin, some of the apparatus and materials. So we need test tubes and the test tube rack, right, in which we will put the albumen and the pepsin. Okay, and then we have the syringes here, one for albumen, one for pepsin, right? And one for the acid, one for the alkaline. So this is our albumen suspension. So you can see it's cloudy and it's got strips of white tissue substance inside also, right? Then we have 1% pepsin. This is the enzyme. So albumen suspension is the substrate, enzyme is the pepsin, and we have distilled water to create a neutral medium, okay, of pH 7. Now this beaker, large beaker with the tripod stand and the wire gauze and Bunsen burner is for you to set up the water bath, okay, plus thermometer. And then stopwatch to time 20 minutes from the start of the experiment to the end. Now, a teacher will also give you pH paper. Usually the pH paper color is something like this, orange, yellow, like that, right? Sometimes the pH paper will come as in a packet. This one is as a reel, a roll of pH paper which you can tear out. This is to test the pH of each solution, of each reaction mixture, okay, to confirm what's the pH. And then we need two of these reagents, 0.1 m sodium hydroxide and 0.1 m hydrochloric acid, to create the alkaline condition, okay, and the acidic condition for the reaction medium. So in some schools, they may have the more advanced equipment, such as the water bath that's electronically controlled. Okay, you just set the temperature here, and it's like a digital water bath, and then the temperature will be maintained. No need to use Bunsen burner. Okay, so it depends on what the school provides. Right, so studying the effect of pH on the activity of the enzyme pepsin. Procedure. Okay, let's look at the procedure. Prepare 3 test tubes and label them P, Q, and R. Then put in 5 ml albumin suspension into each test tube using a syringe. So that is your fixed variable, 5 ml of albumin suspension into test tube P, Q, and R. Next, add the following solution into each test tube. Into P, we add 1 ml of 0.1 m hydrochloric acid. You can use a syringe to insert the solution, and 1 ml of 1% pepsin solution. Okay, this is the enzyme. Q has 1 ml of distilled water and 1 ml of 1% pepsin solution. And then R has 1 ml of 0.1 m sodium hydroxide solution plus 1 ml of 1% pepsin solution. So here we see the manipulated variable, okay, is handled. That is, P contains hydrochloric acid to create an acidic condition. Q contains distilled water to create the neutral condition, and R contains sodium hydroxide solution to create the alkaline condition. Okay, so dip a piece of pH paper into each test tube and record the pH of the mixture, right, by using the scale shown on the pH container. Okay, by referring the color, once you dip the pH paper into the solution, then the pH paper will be of a certain color. So you compare to the chart, okay, to decide what's the pH of the reaction medium. Okay. Now, so this can be taken as a precaution. That is to find out the exact pH of the mixture, right? Next, so you should get one for P. You should get a pH which is less than 7. Q should be around 7, pH 7, and R, the pH should be more than 7. Okay. So put all the test tubes in a water bath with a fixed temperature of 37 degrees Celsius for 20 minutes. Again, this is your fixed variable because the temperature is fixed at 37 degrees Celsius. Next, observe the state or condition of the mixture at the beginning of the experiment and after 20 minutes. Okay, so that's your R rate, the state or the condition of the mixture. That means the clarity or turbidity of the mixture at the beginning, and then compared with after 20 minutes. Okay. So at first, all the mixtures in P, Q, and R will be all cloudy because of the albumin suspension that was put inside it. Okay. Now, after 20 minutes, observe again the condition of the mixture, whether it's clear or turbid or cloudy, and then record the results in a table. So this picture shows us the apparatus setup for the activity 5.2, which is to study the effect of pH on the activity of the enzyme pepsin. Here we have 3 test tubes that have been filled with the albumin and pepsin, the substrate and the enzyme. The only difference is that in these test tubes, the pH are all different, right? For P, we insert in hydrochloric acid to create an acidic condition. For Q, we put in distilled water to create a neutral condition, and for R, we put in sodium hydroxide solution to create an alkaline condition. Then all these test tubes are immersed in a water bath at 37 degrees Celsius for the period of time of 20 minutes. And then after that, we observe the condition of the mixtures, okay, and compare it to the condition of the mixture before or at the beginning of the experiment. So here are the results. Okay, so at first, if you look at P, at first it's cloudy, right? Just like Q and R, at first it's cloudy. But what happens after 20 minutes? Okay, let's observe this animation. Just look at test tube P. So if you observe it over a period of 20 minutes, it will slowly turn clear. So in acidic medium, with a pH of 2, pepsin digests the cloudy albumin suspension into clear polypeptides. Okay, so when you look at the test tube, you can see through the solution because it's a clear solution. All right, now this is the final result, right? Albumin plus pepsin plus HCl forms a clear solution. Whereas for Q and R, they are still cloudy, okay, as at the beginning. So like there's no obvious change, right? So what does this tell us? This tells us that there is no hydrolysis or breakdown of the albumin, right, into polypeptides. That means the pH is not suitable, it's not the optimum pH for the action of the enzyme pepsin. It's not the optimum pH for the enzyme pepsin, okay? So overall, these are the results for the experiment to study the effect of pH on the activity of pepsin. For test tube P, the mixture turns to form a clear solution after 20 minutes. But for Q and R, both mixtures are still cloudy after 20 minutes. So here are the results in a tabulated form. So on the left, we have the test tubes, right, PQR, and the pH is written also on the left-hand side. This is the manipulated variable, it's written on the left-hand side. And then we have the condition of the mixture, that is the clarity or turbidity of the mixture, is the RV, and it's written on the right-hand side, usually, okay? So for test tube P at pH of 2, the condition of the mixture at 0 minutes is cloudy, but after 20 minutes, it becomes clear because the pepsin has digested the albumin, cloudy albumin, into polypeptides. Okay, and the enzyme is active at a pH of 2, and it works most efficiently at a pH of 2. For Q, pH is about 7, neutral, so it remains cloudy, okay, even after 20 minutes. And similarly for R2, it remains cloudy even after 20 minutes, okay. And we know that these two pH, 7 and 8, pH 7 and 8, are not the optimum pH for the most efficient activity or reaction of pepsin, whereas pH 2 is the optimum pH for the most efficient reaction of the enzyme pepsin. Okay, discussion. Why is it necessary to incubate the test tube in a water bath at a temperature of 37 degrees? Because 37 degrees is the optimum temperature for the fastest activity of the enzyme pepsin. Secondly, what is the effect of the pH on pepsin action on albumin? What's the effect of pH? Okay, so pepsin functions most quickly and efficiently at a pH of 2.0 and hydrolyzes the albumin at this pH, right? Whereas at a higher pH, like pH 7 or 8, the activity of pepsin decreases and it does not hydrolyze albumin efficiently, or maybe there's no hydrolysis of albumin, no hydrolysis of albumin. Now, discuss the results obtained for test tubes P, Q, and R. So in test tube P, the mixture changes from cloudy to clear after 20 minutes, right? Then discuss means you have to explain why. So this is because at the optimum pH of pH 2, pepsin functions efficiently and quickly to digest the albumen, the cloudy protein, into clear polypeptides. Now for Q and R, the mixture remains cloudy as before because the pH is too high, not suitable for the action of pepsin. Alright? Now if you're going to explain more, why is it not suitable in terms of what happens to the enzyme, okay? Actually, the change in pH value of the mixture has already changed the charges, okay, which is reliant or depends on the presence of the ion H positive, okay, hydrogen ions. So the change in pH value, maybe to pH 7 or 8, has changed the charges on the active sites of pepsin and substrate surfaces. So maybe both of them now have the same charges, okay. If both of them have a lot of hydrogen or less hydrogen ions and more of other ions, they may be both, the active site of pepsin and the substrate surfaces may be both negatively charged. Then they cannot bind to each other, okay. That means the substrate molecule cannot bind to the pepsin molecule, right, to the molecule of pepsin at the active sites, because both are positively or both are negatively charged. Thus, the enzyme substrate complex cannot be formed, and there is less or no reaction because there's no binding between the substrate and the active sites. Okay, so the enzyme substrate complex cannot be formed, then there's no reaction to break down the substrate to form the polypeptides. So conclusion, the hypothesis is accepted. pH 2 is the optimal pH for the reaction of the enzyme pepsin. Okay, now let's look at some questions related to this experiment. All right, paper 3 questions. All right, so let's try to apply our technique of answering paper 3 questions to this experiment. Let's say you have done the practical test and these are your results, right? So in the real SPM practical test, they may modify the experiment a bit and they may not even tell you which solutions have what pH. pH paper may not even be given. Okay, that's for you to find out the results from the results of the experiment to make your own conclusions, right? So they can modify the experiment. But let's say that these are the results of the experiment. Okay, now then the questions will be asked, right? So the first question is like this: carry out the experiment to investigate the effect of pH on the activity of pepsin, right? So state the observation for test tube P here. So based on these results, can you write down a good observation? So pause the video and try the question first. And we will discuss the answer in a short while. All right, let's look at the answer. So this is a good observation. In test tube P, start from the left-hand side here. In test tube P, when the pH is 2, right, follow from the table, the condition of the mixture, okay, this is the title here, the condition of the mixture, in brackets you can even put clarity and turbidity of the mixture, changes from cloudy, okay, at 0 minutes to clear after 20 minutes, correct? Right, so that is your observation. And here we try to check whether we have all the components. Now, the manipulated variable and responding variable must be in the observation, plus any readings or observations, okay, based on the experiment. So let's look at our answer. In test tube P, okay, we mentioned test tube P, when pH is 2, right? So your MV is the pH, right? The one tick there. The condition of the mixture, so that is your RV, the responding variable. Actually, it's the sub RV, correct? The operational RV that you obtain from the experiment. Changes from cloudy to clear, that is your reading or observation. There's no reading, there's no measurement made here, but you observe that it changed from cloudy to clear. Okay, so that's the observation. After 20 minutes. Okay, so if you have these three components, MV, RV, and the reading, then you get your 1 mark. Okay, or MV, RV, and the observation, what you observe with your eyes. Okay, get 1 mark. Next, state your inference for B1, or you may be asked, explain your observation in B1. Okay, they are both the same things. So an inference is an explanation or a reason for the observation. Why is the observation like that? Okay, so we have already stated the observation. Now let's see how to write the inference. So try and do this yourself first. Try and answer this question, and we'll discuss in a short while. So think about the components for an inference. All right, you must be able to explain it. Explain the observation that you made in B1. Let's look at the answer. So referring back to our observation in B1, at the optimum pH, which is pH 2, we said that the condition of the mixture changed from cloudy to clear, right? So what's the reason for that? It's because the enzyme PEPSIN is most active, right? So no need to mention again that the condition of the mixture changed from cloudy to clear because the enzyme PEPSIN is most active. Okay, no need to mention the condition of mixture. Just straight away explain why. At pH 2, which is the optimum pH, the enzyme PEPSIN is most active, all right, because this is a suitable pH for it to react. And the pepsin then digested or hydrolyzed the albumen, the cloudy albumen, okay, into polypeptides, which form a clear solution, right? So we have to mention that the albumen is a cloudy suspension and the polypeptides form a clear solution. That's why there is a change, okay, in the turbidity of the mixture from turbid or cloudy to clear, right? So 2 marks for this, all right? So for inference, make sure you explain well what happened, okay, for a particular observation or reading, all right? And usually you have to refer back to the objective, which is to investigate the effect of pH on the activity of pepsin, okay? So we can say that at that pH, the activity of pepsin is more, that pepsin is more active, right? At this pH of pH 2, okay? And then the reason why, you have to base it on your biological knowledge, okay, that you've studied about enzymes. Next is to state the hypothesis for this experiment, 3 marks. Okay, so you can try the question first. Remember, hypothesis must have, again, 3 components. Okay, as you see here, let's look at the requirements for hypothesis. MVRV plus correct relationship. Okay, so try this first, and this question first, and we'll look at the answers afterwards. All right, let's look at the answer. pH 2 is the optimum pH of pepsin, at which the reaction rate of pepsin is the highest. All right, now where is your MV? Your MV is the pH, right? So as long as you mention pH, and then your RV is the activity of pepsin, or the reaction rate of pepsin. Okay, so, and then your relationship is that pH 2 is the optimum pH at which the reaction is the highest, at the highest rate. Okay, so we must have pH 2 and then highest rate. Okay, so that's MVRV and correct relationship. So here I'd like to remind you, actually there are 2 different formats here that can be used for hypothesis. The most common one is as MV increases or decreases, RV increases or decreases. Or similarly, the higher or lower the MV, the higher or lower the RV. Okay, where MV and RV is mentioned, and then increase, decrease, all these other relationship between MV and RV. Okay, but the one we are using here for pepsin, the hypothesis for this experiment, is using the second format. Okay, for a few items that are compared, like fruits or food samples, or in this case the pH, we can write this format. Of all the MVs, a specific MV has the highest or lowest RV compared to the other MVs. Okay, now actually then in this case the hypothesis is of all the pH, like pH 2, pH 7 and pH 8, right? pH 2 is the optimum pH of pepsin, right? And that it is the one where the reaction rate, that means the RV, is the highest. Okay, So this format is similar to the second example here, this format, okay? We cannot use as MV increases or decreases, RV increase or decrease. Not so suitable. So it's using the second type format. So memorize this hypothesis for the pH. When pH is investigated, right? The effect of pH is investigated on the reaction rate of pepsin or even amylase. Now for amylase, the experiment is slightly different, okay? To investigate the effect of pH on the activity of amylase. And that one, I hope to come up with another video later on, okay? Where I will give you a question and you have to answer, find out the hypothesis, the objective, inference, hypothesis, etc. Like this one here. Now, fourth, define operationally. So this is also a common question in paper 3. So let's find out how to answer this. Based on the results of this experiment, define operationally the hydrolysis of albumin, okay? Or you can be asked, give the operational definition. Also the same thing, give the operational definition of a term, okay? Like hydrolysis of albumin. See, state, and if you look below at the table here, state the operational definition. Okay, it's the same as define operationally. So the word operationally here means that based on the experiment in the lab. Operationally means experimentally. Based on your experiment that you carry out in the lab, you're asked to define the hydrolysis of albumin. Okay? Right, just a recap now. For operational definition, you must have the basic definition first, and then include the organism, and then you use the basic theoretical definition and modify it to the practical, to the experiment. Okay, modify it based on the experiment. All right. So you must mention the RV by using this phrase. First, the definition, and remember to state the organism if there's any, or the substances used, and then RV, you can put which is shown or determined by the RV. Okay. Now CV may be added to the definition, but so far it is just an optional thing, right? That means this may not be a compulsory requirement, but you must have RV and MV. Now for MV, we can say that RV is affected by MV. Must use the word affected by. Okay? And also make sure that your RV is the sub-RV that you measure or observe in the experiment, not the final or main RV, such as activity of pepsin, or rate of reaction of pepsin. Okay, we don't want that. We want the one that we measure in the lab or observe in the lab, which is the condition of the mixture. Okay, in this case. Now, so the short, the acronym for operational definition is DOREM. Think of the word Doraemon, right? So the cartoon character. Now D is for definition, O for organism, R is for RV, A is for affected by, and then M is for MV. Okay, so DOREM. Think of DOREM. Okay, try and write down your own operational definition for the hydrolysis of albumin. Right, let's look at the answer. Now, the hydrolysis of albumin is the breakdown of albumin by the enzyme pepsin, and it is shown or it is determined by the condition of the mixture, that means the clarity or turbidity of the mixture, of albumin and pepsin after 20 minutes. The hydrolysis of albumin is affected by the pH of the reaction medium. Okay, so let's see whether all the components are here. The hydrolysis of albumin, so you must start with the term that is being asked. Okay, you must start with the words the hydrolysis of albumin as stated here. Okay, so the hydrolysis of albumin is the breakdown of albumin. Hydrolysis means the digestion or breakdown of albumin, correct? So that's the basic definition. By the enzyme pepsin, so mention the substances involved, the enzyme involved, okay? And it is shown by, or it is determined by what? RV. So RV is the condition of the mixture, that means the clarity or turbidity of the mixture, which consists of albumin and pepsin, right? So that is your sub-RV, right? after 20 minutes, so I put in the CV after 20 minutes. 20 minutes is the CV. So that may be optional, but I prefer to write it down. The hydrolysis of albumin is affected by the pH of the reaction. Okay, now affected by, right? So you put it in. So all the components of the operational definition is in. Okay, now you can see the word DORAM here, or DORCAM. If you add in the C for constant variable, then it's DORCAM, like the door camera. Camera they put at the door. So, or if not, you can put it as DORAM. Okay, let's see. Now D, I use the word DORCAM. Okay, now D is for definition. You have the definition here. And then O is for organism, or S, the substance, such as the enzyme pepsin. And then your R is for RV, so condition of the mixture, it's a sub RV. And then C is for CV, okay, constant variable. And then A is for affected by, and M is for the MV, right? The last one, pH of reaction medium is your MV. All right? Okay, so try to remember the acronym. I think I would prefer DORCAM. But if your teacher teaches you has taught you to use without the constant variable and you feel safe, then you can use DORAM. Okay? So DORCAM or DORAM. Now next one. The experiment is repeated for the test tube P, but boiled pepsin solution is used instead of freshly prepared pepsin solution. That's not boiled. Okay? Predict the results after 20 minutes and explain your answer. Okay, so this is a question on prediction. So try to answer it yourself first. And we'll discuss the answer first. Right, let's look at the answer. So in this case, when you predict, you have to compare with the test tube P, the original test tube P. All right, which uses the freshly prepared pepsin solution that's not boiled. But now in this second experiment, you boiled the pepsin solution first. So is there any difference to the results after 20 minutes? Will the mixture turn from cloudy to clear, or will it not? Okay, so the answer is that the mixture of albumin and pepsin will remain cloudy even after 20 minutes, right? Why? So your explanation will get you another 2 marks, right? Or 1 mark. So when the pepsin solution was boiled, E stands for explanation. P stands for prediction. Okay, the P here is for prediction, and the E is for explanation. So when the pepsin solution was boiled, the enzyme was denatured or became denatured, right? Now what is denaturation? As we discussed in the previous video on amylase, in denaturation, the heat is applied to the enzyme, right? The chemical bonds in the enzyme, okay, look at this comment below. Chemical bonds in the enzyme molecules will break due to the heat, and then the active sites in the enzyme molecules will change shape, okay? So they can no longer bind to the substrate. So the rate of reaction will be reduced, even until 0, no reaction. Okay, so here I write active sites on enzyme molecules change shape. So now the shape of the enzyme, the active site, the shape of the active site is not complementary to the shape of the substrate anymore, right? So there is no binding between the substrate and the active site on the enzyme molecule. So no reaction occurs. So pepsin will then be unable to hydrolyze the cloudy albumin because it's denatured. So it will not form any clear polypeptides. Thus, the mixture of albumin and pepsin will remain cloudy with the albumin still present in the mixture after 20 minutes. Now, lastly, you can also be asked to state the variables in the experiment and the methods used to handle them. Okay? Sometimes it's not in the form of a table, it's just in the form of a question. Okay? But here I put in the form of a table, so it's easy for you to see how to answer it. Okay? So any of the variables can be asked. Manipulated variable, responding variable, or fixed variable. Okay? And you may be asked the method to handle them. Now, what is the manipulated variable here? As we know, it's the pH, right, of the reaction medium. Now, what's the method used to handle the variable? Think of the words use different something. So use solutions of different pH, such as hydrochloric acid, distilled water, and sodium hydroxide solution, okay, based on the experiment that you just did. Now, for some other experiments which use different pH, they will give you buffer solution, as written here, buffer solution. So buffer solution is a solution that has a specific pH. For example, buffer solution with pH 7, buffer solution with pH 2, buffer solution with pH 8, and so forth. So if buffer solution is used, then you just write use buffer solutions of different pH, such as buffer solution pH 2, buffer solution pH 7, and buffer solution pH 8. Okay, so sometimes buffer solution can be used in place of all these acid like hydrochloric acid or sodium hydroxide. Okay, they can just give you a buffer solution with a specific pH. Now, responding variable. Always choose the sub-responding variable that we are using in the lab, that you are measuring in the lab or observing in the lab. Okay, so in this case, it is the condition, that means the clarity or turbidity of the mixture, right? Now, method to handle the variable is observe and record the condition, that means the clarity and turbidity of the mixture after 20 minutes. Okay, now the keyword here is observe and record. We cannot say measure and record because there's no measurement made. Okay, for other experiments, it could be measure and record. All right, for example, measure and record the time taken for the iodine solution to remain brownish yellow. Okay, that is for the experiment to study the effect of temperature on the activity of amylase in the previous video. Okay, we measure and record the time taken for the iodine solution to remain brownish yellow. Okay, but in this case, it's observe and record. But you must have both words. You must have the word record here also. All right. Now, is there any instrument or apparatus that we use? Nothing, right? We just use our eyes, so no need to mention by using anything. But if there is a particular apparatus used, then you have to put by using something. Okay. For example, measure and record the time taken for the iodine solution to remain brownish yellow by using a stopwatch. Okay. Right. Next, fixed variable, temperature of reaction medium or temperature of water bath. Okay. So for fixed variable, we must write the word fixed, as if we can write it inside. So fix the temperature of the reaction medium or the water bath at, now if there's a particular value, you have to state it down for fixed variable. So at 37 degrees Celsius. Now sometimes this experiment can be carried out at room temperature. All right. So it can be modified to be carried out at room temperature without using water bath also. Okay. So if it's at room temperature, then you put fix the temperature of the reaction medium or water bath at room temperature. That's all. Okay. Now here is using water bath at 37 degrees Celsius. So to summarize, you have to know the keywords to use when you are explaining the method to handle the variables. Okay. For MV, we also write use solutions of different something or use different something. Use different MV. Then you can also state such as and then give the values for the MV or the variations for the MV. Now, for responding variable, please remember to write observe and record or measure and record. Okay? And the RV, measure and record the RV by using an apparatus. State the apparatus if you use an apparatus. And then for fixed variable, the keywords are fix something at a particular value. Okay? Right, so that's all for this lesson. Thanks for viewing, and please do share, like, and